Hey everyone, we're going to go over Lab 6 and Lab 7. So you should be on the hand washing and normal flora lab. This one you need to make sure that you have your agar plate from Lab 5 because you poured 2 plates and then one you used for the antibiotics and then one you saved for a later date. This particular experiment can take up to 2 days for growth and then maybe a total of 3 days to complete. And then for Lab 7, you need the broth culture from Lab 2, so your skin broth culture, and then this experiment can take up to 4 days to complete. So we're going to go ahead and click on the link to take you directly to the lab experiment. You've got some, background information here about, normal bacteria flora that's typically found on the human body. Typically don't cause any diseases. And then you will be looking at resident and transient flora, as well as, okay, why do we basically, use hand washing as a means of controlling the spread of diseases. You got your questions that you're going to answer, each time at the bottom. And then going into the experiment, the materials you need is that agar plate that you poured. So it's just a plain agar plate. There's nothing, special about it. You need to, have your other information, your other materials here, nothing that you haven't really seen before. And then you've got your lab kit, lab supplies that you need. So essentially what you're going to be doing is you're going to be performing a gram stain. So you need to make sure you have these slides, that you have each solution that's listed here, your inoculation loop, the staining tray. You definitely need the candle because you're going to be flaming, the loop and the lid of the containers. So going into this particular experiment. So this first part, you're just going to take that plate that you poured, and one side you're going to label pre and the other side you're going to label post, and you're going to have a line across the middle. So on the open pre side, you're going to take your index finger and push it for approximately 3 seconds. Be gentle. You don't want to push through the agar itself. And you definitely don't want to have washed your hands before this. So no washing of hands. Just normal, finger that, you've been kind of using throughout the day, basically. Then after you remove your finger, you're going to wash your hands thoroughly with soap and water. Dry your hands with the paper towels, and then on the post side, same finger, press it for 3 seconds, close the plate, and then, incubate the plate for 48 hours. So typically what you should see is on the pre side more bacterial growth than on the post side. So if you don't see anything, you're going to probably have to repeat the experiment. You are going to have a hypothesis. So if you go down here to your data table, you're going to write your hypothesis based on, what do you think is going to happen. You're going to have a photo of your agar plate after it has incubated. So this is the after photo, essentially. Then you're going to count the number of colonies and then look at the morphology. So it's shape, this is the examples of what you can see, elevation, size, all that good stuff. And so when you get to your data table, you're going to count the number of colony numbers, and you're going to use the description, right? So you're going to say, okay, it's, a circulated, curled, convex, small size, right? Something like that in this area right here. Then you've got questions to answer. And then we're going to move into the second experiment. So we're going to come out of here. So this is where you're going to do gram staining. This one, you definitely want to read through the procedure very, very carefully several times beforehand. Gram staining is very finicky to do correctly. So first thing you're going to do is after you disinfect everything and you've got your, protective equipment on, you're going to have a disposable cup with distilled water, another cup with alcohol, you're going to take a blank slide and label one end of it with with the permanent marker. And then you're going to flip the slide over and draw a little circle on the underside of the slide. Sterilize your loop. Okay, now remember, after you sterilize it, make sure you shake it and dry. It's completely dry so you don't kill the bacteria. You're going to take that inoculation loop, put it in the distilled water, and then just transfer a little bit onto that circle, or kind of like in the middle of the circle. You do not want to put it on the side that you drew the circle, because the water can mess with the permanent marker. So just drop a water on the other side. Okay, sterilize it. Then you're going to collect an isolated colony from the pre-section of the agar plate. So the one where you grew, where you put your finger. You're going to put that colony into the distilled water, stir it a little bit, and put your inoculation loop in bleach. So let that slide dry completely, and then you're going to basically do another slide for the post half of the agar plate. Now, obviously, if you have nothing growing in the post half, then there's not going to be a lot to do, but you still need to go through the steps. Then you're going to light a candle. You're basically going to heat fix the sample by holding the slide, passing it over the flame 5 times. This is really, really important. It allows the sample to stay on the slide after you do all of the staining liquids. After it cools, after each slide cools, you're going to go ahead and stain them. So you're going to take that pre-slide, kind of put it in this little tray, and you're going to add crystal violet. You're going to wait 60 seconds. You're going to gently rinse it with tap water over the tray. Okay, so you don't want to be aggressive with this. You just want to be real gentle with how you let the water rinse it. Then you're going to add drops of PVP iodine. Can't talk today. Wait 60 seconds, rinse. Add decolorizer, wait 30 seconds, rinse. Okay, remember, each time you're rinsing, you're being very, very gentle. Okay, it's not an aggressive kind of pouring of water. The last stain you're going to add is the saffronin. Wait 30 seconds and rinse. And then you're going to place that slide and let it dry. You're going to take a photo of it, upload it, repeat those same steps for the post slide. All right, then you're going to complete your data table. So gram-positive results are going to look purple, where gram-negative results look pink. So you're going to have these questions that you're going to answer. If you look at your data table, you're going to talk about, okay, what is the result? So was it gram-positive or gram-negative in here? If something went wrong, then you need to explain that in here as well. Your photo should be the pre-hand washing and then photo of post-hand washing slides. And then you will go to your evaluation, make sure you've done all the questions, and then you're done with this particular lab. So then we're going to come back out and go to lab 7. This is morphology and staining techniques, so more staining. You need that broth culture, so your skin broth culture. So clicking on this link, okay, you're going to learn about morphology. Okay, so essentially different bacteria shapes. You have, okay, how is it typically arranged? You have information about staining techniques. Okay, so different types of staining techniques. What the staining process is for those techniques. Gram staining theory, so difference between gram-positive and gram-negative bacteria, and how to be able to, see it. And then this is talking about the procedure. Now, you basically just did the procedure whenever you did lab 6. So this is just kind of going into more details about it. Make sure, you answer your questions at the end of each one of these. And then for our experiment, the materials you need that broth culture of your skin sample. You need the broth culture of the yeast. Right? Same things that you always typically use. For your lab kit supplies, you need this Congo red, and then you need those gram stains that you just did, and your inoculation loop and staining tray and tea candle and all that good stuff. So for exercise 1, you're going to use a simple staining technique. So you're going to take a slide and you're going to write direct M on one end. And then you're going to flip that slide over, draw a circle, right? And then you're going to take a swab and you're going to real vigorously scrape the inside of your teeth and gums. Okay, the idea is to get some bacteria knocked loose so that way you can stain it and see it. So you're going to place that swab, smear it using a circular motion. Make sure that it's completely dry, and then you're going to heat fix it similar to what you did in the last lab. Okay, let it cool, and then put it in the staining tray. Okay, put the crystal violet on, let it sit for 60 seconds, rinse. Then you're going to very gently blot the slide. Don't overdo this or else you won't be able to see any kind of stain. It'll go away. So take a picture of that. There's going to be a microscopic image provided to you. And then you're just going to repeat this with the same process, but just write 3 neg M. And in this particular instance, you're going to use your Congo red. So just repeating steps 5 through 7, right? Where you're, you know, getting the sample of your cheek. Okay. Don't do 8, 9, 10, right? Come back down here. So after you smear the sample, that's when you're going to put the Congo red stain over it. And then you're going to get a second swab, scrape the inside of your teeth and gums, place that swab into the drop of the Congo red and like kind of mix it around. And then you're going to take that, take another slide and then kind of just follow these directions down here to kind of spread the dye across. Let it air dry. There shouldn't be any liquid on it. Take a photo of it, and then make observations. So you've got your questions here. If you kind of look at your data table, right? What observations do you see about the morphology, the color? Like what do you see with the direct stain versus the negative stain with the Congo red? As for photo 1, right, that's you taking a picture of your slide. Photo 2, this is kind of like what it should look like, a microscopic image of it. You don't have a microscope, so don't worry about that. Then you've got, your mouth negative slide picture. And then this is just giving you an example of what it would look like under a microscope. All right. So then you've got differential stains. So this is basically staining the yeast sample. Same kind of deal. You've got the circle on it. You're going to get some active yeast culture, place it onto the slide, right? Let it dry, heat fix it. Going to do the same thing, with the body sample broth. So put it on there, heat fix it, let it cool. And then we're going to stain them. So you've got with the gram staining. So you've got your crystal violet, right? Same kind of deal as before, kind of rinse it, do the iodine, rinse, decolorizer, rinse, saffron, rinse, right? Repeat it, take a photo, look at the microscopic images, answer the questions, right? Make sure that you do your evaluation, and then you should be good to go.